Today, I'm outlining an experiment designing and validating an expression vector. The gene of interest, the spike gene, encodes the spike glycoprotein, the largest of four major structural proteins found in coronaviruses. The function of the spike glycoprotein is to determine the virus host range and drive cell tropism. Spike glycoprotein is associated with diseases such as SARS-CoV causing SARS and SARS-CoV-2 causing COVID-19. This experiment aims to express high levels of spike glycoprotein via a bacterial transformation into competent E. coli cells, which then can be used to measure and analyze protein expression. Studying this is beneficial for several reasons, including clinical applications and research advancements, for example, towards the coronavirus vaccine development, antiviral drugs, diagnostic testing, and monitoring variants. The gene will be inserted into a plasmid by digesting the chosen vector and PCR amplicon with restriction enzymes. This then generates compatible ends for ligation. Then the digested PCR product will be purified using gel electrophoresis to remove impurities and extract through a commercial kit. Then the purified PCR product will be ligated into the digested vector using T4 DNA ligase, forming the recombinant plasmid. T4 DNA ligase is used because it's highly efficient at ligating compatible DNA fragments, resulting in a higher success rate for the ligation reaction. The chosen vector is PET28A+, which is commonly used plasmid vector for expression of recombinant proteins in E. coli. This vector was chosen due to its approximate size of 5.5 kilobases. The spike glycoprotein has a corresponding mRNA size of around 3.8 kilobases. The vector has a reasonably large insert size, and consideration has also been taken for any additional sequences, such as regulatory elements. The vector contains a multiple cloning site, a region with unique restriction enzyme recognition sites, allowing for insertion of the spike gene in a controlled and specific manner. The T7 promoter is strong and tightly regulated by the LAC repressor, making it inducible to isopropyl beta D1 thioglycopyranose, also known as IPTG. The T7 promoter is recognized by the T7 RNA polymerase, enabling efficient transcription of the inserted spike gene. The backbone of the vector contains essential elements needed for replication in E. coli, including the origin of replication, COL-E1, and kanamycin resistance gene as a selective marker, which allows for selection of E. coli cells containing the plasmid. A T7 transcriptional terminator sequence is used to ensure the correct termination of transcription after the inserted gene. The vector also adds a polyhistine region to the C-terminus of the expressed protein, which can facilitate purification of recombinant proteins. Protein expression from the vector is inducible with IPTG mimicking the natural inducer of the LAC operon, leading to induction of the T7 RNA polymerase and subsequent protein expression. E. coli cells are being prepared using a commercial kit to ensure competency. Then the competent E. coli cells are transformed with the ligated plasmid using electroporation. Electroporation is being used due to the higher transformation efficiency compared to other methods such as heat shock. Then the transformed cells are plated onto agar plates containing the selective antibiotic kanamycin. Clones will be identified through performing restriction digestion on plasmids isolated from positive colonies. This verifies the presence and size of the insert. Following this, Sanger sequencing will be used to confirm the clones to ensure the correct sequence. Transformed E. coli cells will then be inoculated, and the culture will be allowed to grow in LB medium supplemented with kanamycin. Protein expression can be observed by adding IPTG to the culture and incubating to induce protein expression. These protein-expressed cells can then be harvested through methods such as centrifugation for protein analysis. Purification of the protein using affinity chromatography can be completed based on the hist tag at the C-terminal. Post-purification protein analysis methods such as SDS-PAGE or Western blotting can be used to perform analysis. SDS-PAGE separates proteins based on their size. After electrophoresis, the gel can be obtained with a protein dye such as Ku-Masi blue, and the presence of a target protein bank can be observed. Additionally, Western blotting allows for specific detection of target proteins using antibodies. Proteins are transferred from a gel to a membrane, and the membrane is then probed with antibodies that recognize and bind to the hist tag on the target protein. Following this, research can be completed to develop further insights into the spike gene and spike glycoprotein, but potentially impacting applications in both clinical and research settings. For example, advancing knowledge around vaccine development, antiviral drugs, and therapeutics. Thank you for listening. My references are below.